Today's topic is evolution of sporophytes, and which is described or can be described by Telome theory of Zimmerman. So as you know, in pteridophytes, it is actually cryptogams, vascular cryptogams, non-flowering plants, but it is vascular plants. And how the modern sporophytes, including pteridophytic sporophytes, are developed from rootless, then leafless, dichotomous branch, and it was actually described by Zimmerman with the help of his Telome theory. And here actually, generally, it is believed that the earliest and the primitive sporophytes of the land plants was axial in nature. And the leaves and branches were evolved later by a modification of central stem. The primitive land plants were supposed to have rootless, leafless, and dichotomous branch green stems. So dichotomous branch is like this. So these are the dichotomous branching system. This is dichotomous branching. Dichotomous branching system and green plants. Here, as you know, that pteridophytes is a vascular class, so having vascular system, and in vascular systems, it is the simple vascular system, photosteric type. Some alternate branches were terminated sporangial. So here these are actually terminated sporangial. Sporangial. These are alternated branch terminated by sporangial. And discovered these type of plants were discovered during upper Silurian and Devonian periods. And for example, these are the Rhynia, Podiophyton, Xylophyton, and Asteroxylon. And from these plants, actually some questions are here. So how the present sporophytes means having leaves, branches, roots, and sporangia were developed. And from these type of plants, so how the modern plants actually developed. So among many, Zimmerman, his Telome theory in 1930, it is actually widely accepted. And he published it in Telome theory in Manual of Pteridophytes in 1938. So according to this theory, the Telome theory and the specific features, and these are all the vascular plants were evolved from a very simple leafless and a dichotomously branched ancestral type like Rhynia. Then plants is made up of sterile and fertile branches. And here, so those branches, so which produces, or these are actually the fertile branches. And the others are the sterile branches. So having the fertile branch and the sterile branch. Then fertile branches terminated by sporangia, and these branches, fertile branches, are terminated by like this sporangia. And the simple terminal parts of dichotomy is called telome. So the terminal part, these are called as the telome. These are known as the telome. And the others are actually called as the mesomes. These are telomes, and the other parts are the mesomes. These are mesomes, and the terminal parts are the telomes, and the others are mesomes. And two telomes meet at the point of forking, and this is actually called here, this part. This point is called forking. This is forking. portion of the stem below the forking is called meson. So here is forking and this part meson. Here is the forking, this part mesons. Sterile or vesicular phylums are called phylums. So these are actually sterile phylum. These are also called as phylloids. And sporangia bearing phylums are called the fertile phylums. So here is the phylloids as well as the fertile phylums. So the terminal part phylums, then below the forking is the mesons, then the these are the fertile phylums, and these are the phylloids or the sterile phylum. Then as evolution proceeds, there was a sense from dichotomous to the sympodial branching as a result of the fusion of the two or more phylums called telome traces. So ultimately, C phylums will be. So these phylums they undergo fusions and resulting as a chain phylums. Then the fertile phylums traces. These are again also called the traces, telome traces. When they fuse each other and produce the telome traces. So if these two fuse and this produces this sporangial traces. This is called as the sporangial traces. The fertile telome traces are called sporangial traces, and the sterile, if these two fused, these two sterile telome fused, and these are called the phylloid phylums. Both type of phylums occur together. When here it is actually together, it is called as the mixed phylum, mixed phylum traces or mixed thin phylums. So primitive vascular pictograms originate from green algae. According to Zimmerman's postulation for vascular pictograms. So according to Zimmerman, the primitive vascular pictograms originate from green algae. The unicellular green algae divides in all plants. So here, unicellular green algae divided in all plants. Suppose in parenchyma, all plants means suppose this is unicellular green algae, and it actually divided in all plants. When these plants, or here, the other plants in here. So ultimately, it produced the whole structure. Later the meristematic tissues develop and erect readily constructed branches came to exist. Appearance of the distinct alternation generation coming up, in sporophytic generation and the gametopathetic generation coming up. Then these sporophytic branches dichotomously and possess this central conducting tissue. Ultimately, from this unicellular algae, it divides in all plants and gives rise to the dichotomous branching sporophytic plants. Dichotomous branching sporophytic. So from here, from this unicellular algae, ultimately gives rise to dichotomous branching the sporophytic plants. So these are early vascular plants evolved during the upper Devonian and Devonian periods. They possess no roots, no leaves. They possess root-like rhizomes. So ultimately, here they possess some root-like structure, and these are called the rhizomes. Possess an heteromorphic organism, and these plants have actually heteromorphic, this similar type of organism of generation, heteromorphic type of means gametopathetic generation and sporophytic generation are different. So there are some elementary processes actually through which or by which the present day sporophytic plants are developed or produced, and these are called the elementary processes. And elementary processes means these are overtopping, eliminations, webbing, syngenesis, reduction, and recurrence. So overtopping, so equally dichotomous exists, develop unequal dichotomy, and this resulted in the formation of short and long branches. These short branches appeared as lateral shoots, and this led to the development of the sympodial axis to the monopodial axis. So ultimately, so the dichotomous branching system. So here, so this is dichotomous branching system. Branch, and so these are equal. At first they are in equal positions, and gradually they become like this. So the equally dichotomous axis here, it is produced, it becomes unequal, and later it becomes like this. So here actually sympodial axis to the monopodial axis. Sympodial axis ultimately develop the monopodial axis. Then next elementary process is proplanation in single plant. Then planation SMS. The equal dichotomized, like originally in more than one plane, and these branches become arranged in a single plane. In a single plane, in a single plane, called replanation, and led to the evolution of leaves. And the next elementary process is synthesis and weaving. Then here, actually, what happened? The parenchymalous tissues are developed. Parenchymalous tissues develop in between the telomes. So the adjacent telomes and the mesomes were connected with each other by the development of the parenchymalous tissues, and this is actually called syngenesis. It is syngenesis. Syngenesis development. Syngenesis development of parenchymalous tissues. These are actually parenchymalous tissues, and ultimately it gives rise to leaves. Give rise to leaves. Syngenesis may be two types: the foliar syngenesis, the formation of the leaves, and the axial syngenesis. Then next one is the reduction. It is brought about by the reduction of the telomes to a single needle-like lateral appendage and microphase. So all these things actually reduced and it becomes up to the needle-like leaves, like in Lycopodium and Cellular. Then the last one it is the recurving or the recurvation. In recurvation, during this process, the fertile telomes were supposed to become reflexed. Fertile telome. So here, so fertile telome. Then it cut, become cut like this. This fertile telome. But ultimately, it prefers to the means potential bend downwards as it is sphenospherical. Spherical all differs sporangia and they become downward. And another is the this incarnation, and this led to the shifting of this sporangia to the antral surface of the leaves. In higher type of plants or the pteridophytes, so what we get at the antral surface of the leaves, the sporas or the spores are developed at the antral surface of the leaves, and here like this. So this is all about the development of these sporophytes. In case of muscular cryptographs, means how the presently or the currently sporophytes are developed from the rootless and the leafless dichotomous branching plants. Thank you.